This is a presentation on some of the research work being done at the Institute of Neural Regeneration and Tissue Engineering. Damage to neural tissue is one of the leading causes of death and permanent disability in the world, and it also presents one of the greatest challenges in current medical care. The burdens of neural tissue dysfunction are carried by millions of people who suffer conditions of stroke, traumatic brain injury, spinal cord injury, nerve injury, and neurodegenerative diseases like Parkinson's, Alzheimer's, Huntington's, and amyotrophic lateral sclerosis, or ALS. Every year in the United States alone, there are about 1 million new cases of traumatic injury to the brain or spinal cord, and nearly 1 million strokes, plus 500,000 new cases of neurodegenerative disease. This means well over 10 million people in the United States alone are living with the permanent effects of these diseases. Neurological diseases tend to cause a tremendous amount of long-term suffering, and yet there are only minimally effective treatments that can be provided to patients. There are currently no medical interventions that can directly repair and restore neural tissue structure and function in the brain and spinal cord. The overwhelming problem with neurological injuries is that we have no tools or capabilities to reconstruct and repair the neural tissue itself. But what if it didn't have to be this way? What would be the best way to guide and direct cells to reconstruct functional neural networks? And in particular, how can the intricacy of neuroanatomical pathways and networks be reformed at the cellular and subcellular levels? I'm going to present a unique approach that combines stem cells within a special three-dimensional architecture. For neural cells in particular, three-dimensional culture is essential to reconstruct the innate relationship of structure and function in neural tissue. Furthermore, three-dimensional constructs enable transplantation into an in vivo environment, and also more realistically reconstruct cellular interactions and adherence with surrounding cells and matrix. 3D cultures also better replicate the mass transfer characteristics and diffusion properties of neuronal tissue, which is important for preparing cells for survival after implantation. For example, recent evidence suggests that stem cells implanted into the harsh conditions of the body have better survival if they are cultured and prepared under the stress of less than perfect conditions, a form of preconditioning as it is called. Prior to this work, I had studied neuroprotective and neurodevelopmental cell signaling mechanisms in the nervous system. And one of the things that happens when the brain is forming in the early stages of development is that radial glial fibers, seen here in blue, extend fibers that ascend from the ventricular plate, which is where neural progenitor cells originate, and neural precursors, which are in green, migrate along these fibers up to the cortical plate, where they form the cortex of our brain. Nature is always the best inspiration for design, and this led me to wonder whether this mechanism could be used to organize the structure of engineered neural tissue. And as you'll see in these experiments, I created nanofibers, which appear in blue, and I used neurons, which express a green fluorescent protein and therefore appear in green in the following images. So the hypothesis was that the combination of nanofibers within three-dimensional hydrogels could help replicate the development of neuroanatomical pathways in the brain and spinal cord, especially if the nanofibers were coated with certain biologic molecules that could aid in cell attachment. It seemed that combining mechanical and biological signaling cues into one three-dimensional construct might provide benefits such as guided neurite extension and scaffolding that could serve as an attachment point for the cells. The patterned and functionalized nanofibers serve as a scaffolding, which is embedded within 3D hydrogel architecture. These constructs are made of biocompatible materials that are absorbed and metabolized by the body over a period of weeks to months, and most importantly, these composite constructs have demonstrated the ability to control neurite direction and enhance neurite outgrowth. This work will enable progress in the field of neural tissue engineering, with the ultimate goal being to restore functional neural tissue to patients with devastating neurological diseases, including stroke, spinal cord injury, tumor resection, and neurodegenerative diseases. It will also greatly expand our understanding of neural function and neural networks. The ability to guide and restore neuroanatomical structures with targeted network formation would be one of the greatest achievements of science, and this work will help advance us in that direction. The following work was recently published in the Journal of Neural Engineering, and it is an open access article, so anyone can obtain it for free, and I will just briefly summarize it here. So this is a simplified schematic to show the process of creating electrospun nanofibers. Nanofibers are made from a polymer solution, in this case polycaprolactone or PCl, and that is infused out a nozzle across a high voltage potential. Nanofibers are collected onto a spinning collection drum, and the alignment and the thickness of the fibers can be controlled by a variety of parameters. The fibers are then biochemically modified on the surface with certain signaling protein molecules for which neurons have an affinity. These fibers are then transferred to a culture well and embedded within a hydrogel architecture. Prior to placing the fibers in the hydrogel is cellularized with neural stem cells. Some of the cells fall through the gel and come into contact with the fibers. The hydrogel is also treated chemically to allow cross-linking, which helps stabilize the hydrogel, kind of like Jell-O. And this reaction occurs at physiologic pH, does not produce byproducts, and doesn't involve the cells themselves. The fibers were suspended and embedded in hydrogel by using an external scaffolding to support and shape the entire construct. In these experiments, the constructs were a simple cylindrical shape, but many other shapes and architectural designs are possible. I also experimented with a couple types of hydrogel, including Engelbreth-Holmes swarm matrix, also called matrigel, which is an extracellular protein matrix purified from sarcoma cells and which has a lot of functional groups for cells to grab onto. And then another type of hydrogel is hyaluronic acid, which is a natural component of extracellular matrix in neural tissue and in biologic terms is relatively inert. This is an image from an environmental scanning electron microscope showing the suspended nanofibers. In these experiments, I used 3 different types of nanofibers. First, plain untreated polymer fibers. Second, polymer fibers mixed with collagen, which becomes a gelatin in the electrospinning process. Or 3, polymer fibers that were biochemically functionalized with certain cell adhesion molecules. The average diameter of these fibers was made to be about 800 nanometers. Here is a closer view of the fibers, and the scale bar is 10 microns. It's worth noting that both the PCl of the nanofibers and the hyaluronic acid of the hydrogel are materials that are biocompatible and have already been FDA approved for use in humans. The next image shows that the fibers can also be placed in a grid pattern. The cells had been transfected with green fluorescent protein, or GFP, which is a protein that makes the cells glow green under certain wavelengths of light. The cells were cultured in certain growth factors that induced neuronal differentiation and development, and this image shows standard neural cultures on a 2-dimensional surface. Neurites extend from the cell bodies, and if we begin to outline the neurites in red, this is the typical morphology that starts to emerge. You can note from the scale bar that each neuron cell body is about 10 to 20 microns in diameter. So let's look at some of the more interesting results. Here is a zoomed out microscopic image of functionalized nanofibers in the hyaluronic acid hydrogel, and you can see the scale bar is now much larger at 400 microns. This image shows both fluorescent imaging of the cells and optical imaging of the fibers together. We can see several conglomerated groups of cells or spherical clusterings of cells at multiple depths throughout the hydrogel, and this is expected. These cells don't have much to grab onto in this particular hydrogel. However, once they come into contact with the suspended layer of nanofibers, you start to see a beautiful spreading of individual neurons along the fibers, which we'll see closer magnification in just a moment. So here is the same image, but only with the fluorescent channel, which shows only the cells. One thing that's worth noting here is that it turned out to be very convenient that the cells alone could be imaged in a fluorescent channel, while the fibers could be imaged in a separate channel, since the fibers did not autofluoresce at the GFP wavelength. This allowed these images to be overlapped and analyzed for colocalization, thereby analyzing the length that neurites track directly along the fibers. This video shows the focal plane moving through the depth of the hydrogel, and as the view moves away from the fiber layer, some of the isolated cell clusters come into focus. Tilting across the construct, we can begin to see the very thin neurites that have extended out from individual neurons at the fiber layer. And zooming in a little closer, we can see these beautifully aligned neurites following the fibers and extending between neurons. So after marking the morphology, this is what an image looks like. Red shows the neurites and the cell bodies from where they extend, and white shows where fibers and neurites are co-localized together. If we then add the fiber channel, which is shown in blue, This then combines all components to help understand how this all comes together and what is happening here. So again, cells are green, nanofibers are blue, neurites are marked in red, and neurites that are directly tracking on nanofibers are shown in white. Just from the eye alone, we can see that neurites are orienting in the same direction as the fibers, and that neurites are extending directly along these fibers. Then finally, one last look at the original neurons themselves without the morphological markings or the fibers. This image was really so much more impressive and stunning under the microscope, where you can actually see it in 3D with the two eyepieces and where everything is more clear and vivid. But a two-dimensional image will just have to do for now, since any fancier 3D imaging requires a lot more expensive equipment. There are many ways of quantifying the morphological results, and I'll just present the most important ones here. They are measurements of neurite angles, neurite tracking of fibers, and neurite length and number. The angle of each neurite segment was measured relative to the fiber with which it was in contact, or relative to the nearest fiber if not contacting a fiber, or horizontally to the right of the image if the condition had no fibers. And the length and angle of the segment were recorded. It was important to extract meaningful information from the data. So for example, if one neurite extended in one direction at 0 degrees, let's say, and another extended in the other direction at 180 degrees, then the average of those two would be 90 degrees, which would suggest a result that's opposite of the fact that the neurite was tracking the fiber. So the easiest solution to this problem was simply to measure the closest possible angle from the neurite to the fiber, which essentially constrains the angle to be between 0 and 90 degrees. These graphs represent the distribution of neurites shown in 5-degree increments, and these results show that, as expected for control conditions, neurites spread in random, evenly distributed directions across two-dimensional surfaces, as we see up at the top left, and in three-dimensional hydrogels without fibers, as we see at the top right. When the neurons are grown in three-dimensional constructs with aligned nanofibers, we see a tremendous alignment of neurites with the nanofibers, even with no biochemical functionalization of the surface of the fiber, as we see at the bottom left. When the fibers are functionalized, we see even more significant alignment. A tracking affinity factor was calculated per cell, which is simply a percentage of the total neurite length that is directly tracking a fiber. So a tracking affinity factor of 100% indicates that 100% of the neurites are in direct contact with the fiber, whereas 0% would indicate that none of the neurites follow the fibers. So if we look at the neurites tracking along fibers, we see that on two-dimensional surfaces with plane fibers, we get about 40% tracking of just the plane fiber, which is pretty interesting in and of itself. When using a whole array of different fiber types and different hydrogel types, we don't really see major differences until we get to the functionalized fibers in the hyaluronic acid hydrogels, and suddenly the amount of tracking jumps significantly to 66%. Now, looking at the number of neurites per cell in blue, and the average length of each neurite per cell in red, there are minor differences between most of the groups, but when functionalized nanofibers are used in the 3D hyaluronic acid hydrogel, the neurite length nearly doubles on average compared to plane fibers in either 2D or 3D, as seen on the right. We see that the number of neurites per cell also changes, as shown in blue, and by multiplying the red and blue, we can obtain the average total neurite length per cell for each condition. If we look at only the longest continuous neurite per each cell, these results become even more striking. They show that the functionalization of nanofibers again more than doubles the average length of the longest neurites, and this was after only 4 days in culture. So to summarize, biochemical functionalization of the nanofibers in three-dimensional hydrogels enabled significant alignment of neurites with the fibers, and significantly increased neurite tracking along the nanofibers, and also significantly increased the distance over which the neurites could extend. Altogether, this work demonstrates a novel approach for creating three-dimensional controllable micro-patterned scaffolds for guiding neurite outgrowth of neuronal cultures. Three-dimensional constructs hold a lot of important potential for implantation and grafting into neural tissue, and the integration of nanofiber scaffolding holds important potential for replicating specific neuroanatomical pathways and for guiding neural network formation down to the subcellular level. These constructs would be directly applicable to specific areas of damage that are amenable to graft implantation, particularly things like spinal cord injuries, nerve injuries, areas of cortical damage, or tumor resection sites. This work may also play an important role as a therapeutic approach to neurodegenerative diseases where delivery of stem cells and the reconstruction of functional neural networks is necessary. The combination of nanofiber scaffolding with cellularized hydrogels in this study demonstrates the integration of structural, cellular, and molecular signals into a single construct. This also provides evidence that further fiber functionalization approaches can be implemented in more complex patterning, and it opens the opportunities to investigate the application of intricate axonal guidance signals in 3D constructs. This approach may also be viable for creation of functional neural networks with patterned neural circuitry and the ability to direct neurodevelopment of stem cells. However, the creation of functional neural tissue will still require overcoming many challenges in order to prepare the technology for successful clinical implementation. Neural regeneration is notoriously difficult for a variety of reasons, particularly due to the complexity of the neural environment and the cellular and subcellular architecture. In addition, it is very important to understand the limitations of oxygen and nutrient delivery to cells within these 3D constructs. So these constructs were modeled for diffusion characteristics to ensure adequate supply, and this involves using the diffusion equation along with diffusion properties of the hydrogel and metabolic rates of the cells, but I'll describe all this in another presentation in the future. Suffice it to say that this information all becomes very important when working with the fragility and capriciousness of neural stem cells. Diffusion limitations may dictate size constraints of the constructs and may require additional features such as tiered stacking of the constructs with fluid vents or vascular channels through the hydrogel in order to help circumvent diffusion limitations. Further problems can arise with the integration of neural connections between the implanted construct and the neural tissue itself, including problems with the coaptation of axonal pathways or deleterious side effects of hyperesthesia and hypertonia that might occur in some cases of spinal cord rewiring. Nevertheless, the use of patterned scaffolding matrices may help overcome these problems by guiding neurite extension, by targeting proper neural connections, and by providing a local environment that supports cell survival, proliferation, and differentiation. In fact, recent efforts using the combination of neural stem cells within unpatterned fibrin scaffolds that contained growth factors has demonstrated remarkable ability to extend functional axonal connections across lesion sites in spinal cord injuries in rats. And cross-linked hyaluronic acid hydrogels have shown improved survival of stem cells implanted in the brain. So this further suggests that the combination of cells, scaffolds, and hydrogels may indeed be an ideal approach for regeneration and reconstruction of neural tissue. As I mentioned previously, the ability of neurites to follow nanofibers may be related to the migration and extension of neurons along radial glial fibers early in neurodevelopment. The roles of radial glial fibers and neural progenitor zones play a unique role in the shaping of the human brain, and the ability to study this with human neurons is important not only for regenerative medicine, but also for studying diseases that are unique in humans, especially neurodegenerative diseases, neurogenetic diseases, psychiatric illness, epilepsy, and certain types of brain tumors, and many other conditions. These 3D constructs create an in vitro model of neurodevelopment and of the pathological disease processes and mechanisms. Altogether, this study serves as a proof of concept that three-dimensional neural tissue structure can be assembled with seeding of induced stem cells on functionalized scaffolding. Future work will attempt more complex patterning to replicate neuroanatomical structures of the spinal cord, cortex, hippocampus, and others, as well as seeding with induced pluripotent stem cells, thereby opening the possibility of treating neurological tissue damage with a patient's own cells on scaffolding that replicates the innate neural architecture. The overarching aim of this work is to create 3D neural tissue constructs with more complex pattern control and more detailed molecular functionalization in order to replicate neuroanatomical pathways and functional networks. This work therefore holds important potential as a novel therapeutic approach for many diseases of neural tissue, including stroke, spinal cord injury, traumatic brain injury, and neurodegenerative diseases. That's as far as we'll go for now. This work is dependent on the generous charitable donations of those willing to help in these endeavors, and if you would like to donate to these research efforts, even if it's a small amount, we certainly could use your help, and all donations are tax deductible and go directly to the biomedical research itself. Please visit the Institute's website at neuralregeneration.org or email research@neuralregeneration.org. Thank you very much.